All right, we are talking about protein synthesis, and they talked in the video. Synthesis literally means making. So with protein synthesis, we are making proteins or creating proteins.
Why is yours colorful and theirs is black and white? That's not fair.
I knew somebody was going to ask. Listen, it's because it uses so much ink for me to print off all of your color notes. And then every day you guys leave biology, and I find like 3 of them on the floor.
Okay, so don't print, just print the people that actually do it.
Yeah.
Next time I will. Next time I'll give you guys color copies.
For the people that
Well, but
actually
some people like to color their own too. So
Well, ask.
Do a poll.
Yeah.
Who would like a color copy? Okay,
Me,
perhaps I will.
want one.
So, we're talking about protein synthesis. Let's talk about what are proteins and why are they even important in our bodies. So, proteins are going to be made of amino acids.
What?
Amino acids. Hopefully you know that because that was on that macromolecules web quest, and we've talked about our 4 main macromolecules. Proteins are one of them. So proteins are made of amino acids. That's their monomer. And then they're then joined together in polypeptide chains.
Oh my god. Poly what?
Polypeptide. Anytime I say a word over 8 letters, somebody says something.
I can't turn around, so I hope I
Polypeptide.
Yeah.
3 terms.
Right.
Polypeptide. Polypeptide chains.
Pretty sure I spelled it right.
These polypeptide chains are then folded to give the protein a specific structure. So our polypeptide chains full of amino acids are folded to give the protein a very specific structure. That structure is really important. If our proteins fold wrong, then they can't do their job. So they have to be a very specific shape, and that allows it to carry out its function.
Wow, look at us go.
A lot of genetic diseases that happen are because the proteins fold wrong, and then it gives you diseases. Like, it's not good. It's really important that it folds properly. Now, proteins are a very important component of our cells and bodies. They have a lot of different things that they do for us. Some examples are they have roles in cell transport, so they're moving things in and out of the cells. They do things in our immune systems. They allow us to move with like our muscles and our skeletal system. They do a lot of hormonal responses, and they do even more. So proteins are doing all of the work in our bodies. So it's all of the work. Proteins determine the color of your hair, the color of your eyes, the color of your skin, all kinds of stuff. It's all proteins. Proteins are a lot. Now we'll go over here. Where is protein synthesis happening? So in eukaryotic cells, which are what we are made out of, transcription is going to happen inside the nucleus. So inside of the little brain of the cell, transcription happens. And then the other kind of big step is translation. And translation happens in the cytoplasm, which is kind of the gooey stuff in the cell. So transcription is going to happen inside the nucleus. That's because our DNA can't leave the nucleus. It has to be in the nucleus. Once we make that mRNA, it can travel outside of the nucleus, and it does everything else in the cytoplasm, where there's probably a little more room. So we can draw some DNA in our cell here. We've got like a double helix shape. So if you do one squiggle, and you do an opposite squiggle like that, that's what DNA looks like. And then it's got these little ladder rungs as well. So these are our different nucleotide bases.
Wow, I drew DNA.
So that is what DNA would look like if we zoomed in really, really, really far. Now in the cell, our DNA is packaged into chromosomes. But if we zoomed in really far, this is what our DNA looked like. If we wanted to draw like a chromosome, they kind of look like this. So that's what it really looks like in the cell, with our little chromosomes, these little like bumpy X's.
What?
So this is what it would really look like as a chromosome, but this is what it looks like if you zoom in really far. Either one is fine.
She keeps saying really far, but in all reality, it's really close.
Yeah, I guess that would be true. It wouldn't be like zooming far out. It'd be zooming like far in. Yeah, you're right. I didn't know I was doing that. So then if we label, right, so this purple kind of circle, this is our nucleus. That's the brain of our cell. That's where all of the DNA is held. These little black dots that are floating in the cytoplasm and then also all over our rough endoplasmic reticulum. These are our ribosomes, yep. And the last one is cytoplasm. Nice job.
No, the other one here.
So cytoplasm is all of the goo that everything is suspended in.
Wait, what?
Now, in eukaryotic cells, transcription and translation happen in different spots. In our prokaryotic cells, transcription and translation are both going to happen in the cytoplasm. And you'll notice that if we look at our prokaryotic cell, there is no nucleus. So our prokaryotic cells don't have a nucleus. Their DNA is just like loose spaghetti in their cytoplasm. And so transcription and translation can happen just in the cytoplasm because the DNA is not held in a separate area. This is going to happen in the cytoplasm. Now we call this whole process, it has a special name, it's called the central dogma of biology. And I have not used that word yet, but it's called the central dogma of biology. Overall, the central dogma means DNA to RNA to protein. That's all you have to remember. Okay? Riley, how many words do you think you say a day?
Huh?
How many words do you think you say a day?
Rough estimate?
Too many.
1 million.
Too many.
An hour.
Zip.
What was the next weirdest word Dylan has said to you guys?
He said one yesterday. No, not yesterday, 2 days ago. It was something along the lines of these molecules of anger. They can't float away. Something like that. It was really weird. I keep track of all the weird things Dylan says in a document. He's aware of it. I've got like 5 things in there now. Okay, so this is the central dogma of biology, where we go from DNA to mRNA to protein. And we do that through transcription. Where we are taking DNA and making our mRNA. With transcription, the DNA code is transcribed into our mRNA strand. Now, they talked in the video, and I also mentioned it yesterday. If you remember that transcription comes before translation, right? C will become before L, so we'll write translation here. So this one has an L in it, this one has a C, and in the alphabet, C comes before L, so transcription is going to come before translation. That's how I remember it, because it is easy to get these two flipped around. I never understood the order until I was in college, I'll be honest. So, transcription, C, becomes before translation, L. Okay. Now with translation, our mRNA strand is translated into a protein. So with translation, we have our mRNA strand that becomes a protein.
Wait, what? You know what? I really hope that you fall off the tree. What? Hey,
Riley, you got 5 pennies?
what's your name? What's
Okay,
his
can
name,
you
Steve?
please stop doing that though? Like it's
Why are you leaving what I was writing, man?
Write faster.
two words.
What is
Riley, it's right there.
Alright, I'm writing it, home.
Also, it's literally in the picture. mRNA is translated into protein. Okay. Now, our DNA is the genetic code.
What's the what?
Genetic code.
Okay. Chill out, you don't gotta get all upset, Skitty.
So it's the genetic code that determines the genetic characteristics.
How was your spaghetti?
Characteristic.
Actually, so good. Thank you for
Yeah?
asking. Yeah.
Did you like make it good, or who made it? You
I didn't
or-
make it. Tristan made it.
Yeah.
but yes, it was
did you
delicious.
did you go home and it was already made?
No, he wasn't home yet.
That's kind of.
I went home. I took a shower. I laid in my bed for an hour, and then he made me spaghetti. That's how my night went. And then I made a Jeopardy
I
PowerPoint.
know you're mad that I didn't show up, but like.
I'm not.
This
I'm not.
weekend, I promise. No, not this weekend. Next weekend, because me and Perry and I'm pretty sure Logan are staying at the hotel.
What
at
hotel?
Soyigo.
Why?
Because I get free rooms all the time, so I don't really use them. So my mom said I can have this one.
Good thing I live nowhere near. Please
Good
don't come
thing
to my
that we
home.
have a truck.
Don't come to my home.
I will be there.
We'll find a way not to be
Like
there.
7 p.m. sharp.
Okay.
And you do know,
I'll be gone.
I followed the rules by Texas. You come on my land, I shoot your ass.
She lives in an apartment, so she can't do that.
No. All right. Sections of DNA that code for proteins are called our genes.
Oh.
Different than the jeans you wear on your legs.
What? It was so random.
What?
That's why I love Kentucky.
So the shoot.
Okay, well we're in Michigan, so why are you?
Do you know what
talking
it's
about
called?
Kentucky?
What?
The law? Stand your ground law? That's what you're talking about. The shoot your.
The shoot law.
Okay.
Oh,
Okay.
the shoot law.
So DNA cannot
Can you
create.
stop judging me?
I'll judge you every day.
I feel harassed.
DNA is not able to create proteins on its own, so it requires
That's all I'm saying.
it requires the help of mRNA. We cannot just go from DNA to protein. Proteins have to be made in the cytoplasm. DNA cannot leave. Also, mRNA is a single-stranded molecule, and the single-stranded molecule allows the riboplasm to read it.
I'll call you over and stand my ground.
Or ribosome, not riboplasm. What am I saying?
What?
Ribosomes. To read it. So mRNA is a single-stranded molecule that's going to be built using the DNA template.
It's not a law.
It is an emphasize.
Now, when we create mRNA from DNA, we call this process transcription. After transcription, the mRNA then leaves the cell. It goes through a ribosome, and it's used to build a protein in a process called translation.
Can you shut the fuck up, bro? Yeah, well, it's kind of
Bailey,
like
what's the next page? The
scary. I'm
transcription.
trying to figure out what the
transcription
point
one?
is so that I can like,
Transcription is next, yep. Because we go from transcription then to translation. Okay.
You don't have to kill Perry.
Okay.
Harry would just be like, I'm not watching. he just looked the other way and just hold his phone like this.
So with transcription, we are taking DNA and we are changing it to mRNA.
No, Harry's grandma fell. She was at the hospital for like that.
So with transcription, we're taking our DNA, we're turning it into mRNA.
Okay, I don't think they
You told
care
me the
about
story.
no grandmas because Ava literally signed a petition to euthanize people over the age of 60.
That's
Okay,
true.
what is it? A little arrow for me?
DNA to mRNA.
Yeah.
Okay. Now we're talking about transcription. How does transcription even work? So DNA is unzipped.
Nice people over 40 turning 40 over the
Don't euthanize people over 40, bro. My dad's over 40.
No, I'm not gonna murder everyone's parents. You won't
Bro.
murder everyone's.
My mom's 39, so she's good.
Okay.
Oh, wait, my
My
dad.
mom's 30.
That's how you gotta go.
Our DNA is unzipped, so its bases are exposed. After DNA's been unzipped, RNA polymerase.
Polymerase? What?
Polymerase.
RNA polymerase or RNAP. So transcription is very similar to how we're like recreating DNA. So we talked a little bit about how DNA is recreated and copied. This is a very similar process.
Yeah.
Yeah.
What's the next one, man?
It attaches to a promoter sequence.
What?
promoter.
Oh.
Sequence of DNA, and it begins to add complementary RNA nucleotides one at a time.
Does it have to be one at a time?
Yeah.
Like if it's 4 at a time, is it bad?
I mean, it goes really, really fast. So it's like one at a time, but like super fast. So
So what if it's 4 at a time, but like...
Bailey, I can't wait to watch this. Like this whole playback bag, I can't.
it'll be interesting.
I
It builds up a strand of.
it posted on Google Classroom.
Are you serious?
I'm dead ass.
Bro, this is great. I
It's
love
posted
it.
on YouTube.
It posts on YouTube too, apparently.
Ah, great. Can I like quote it, dude?
What?
Can I quote something?
What does that mean? Like
Huh?
in daily life?
Do you know what a quote is? Like,
Yes, I know what a
I'm
quote
sorry,
is.
I forgot you didn't major in English.
I did not. Thank goodness. We can call messenger RNA mRNA. So mRNA is what it's shortened to.
Dude, people don't even watch your YouTube for fucking content. They just watch it for the background noise.
Probably. In eukaryotes,
No problem.
the mRNA has non-coding regions.
Not those fucking people, bro.
It has non-coding regions or regions that don't like really do anything. These are called introns. Introns. So these are areas where we copy it, but then we realize like, oh, we don't need that. So then we get rid of them. So they are removed in a process called splicing. Which sounds kind of violent. It's not. It just cuts out the parts that we don't need. And then the mRNA leaves the nucleus through nuclear pores. So the nucleus has these little like gaps, and the RNA will leave.
It doesn't say through nuclear, it says through nuclear.
It sure does. It sure does say nuclear. It's supposed to say nuclear. I didn't make this.
Wait, where?
Before it pour, it says nuclear.
Oh my gosh.
Nuclear.
I don't know if I noticed that.
Oh well. So our double-stranded is our DNA. So that is our DNA. We can see here that our DNA has been unzipped, right? So we've opened it up so that this mRNA can fit in here. So this is our mRNA. This other kind of green strand is the mRNA. So it's just a single mRNA.
Oh wait, where are we at?
We're labeling.
Okay.
So DNA is our double-stranded. Our mRNA is this single strand. It doesn't have a double. So it matches up with these guys, and then we create complementary bases. Now this big kind of comb shape, this is our RNA polymerase. So the RNA polymerase attaches to the DNA and the mRNA, and it helps read it. Now we group our bases in groups of 3s. So we call these triplets. Or sometimes you'll see the word like codon. So I'll put that on there as well.
Codon?
Codon.
What?
A triplet or a codon. So our bases are always read in groups of 3. We don't read like a single one at a time. In our bodies, they're in like little chunks of groups
So
of 3.
you gave us a test or quiz. What word would you put? Would you put triplet or codon?
I would probably put both. Since you have learned both, I would list both. I would put one and then I put the other one in parentheses.
Dude, I forgot we were gonna have this as a pet.
Yep. Now the strand that we are copying on our DNA, this strand that we are copying, we call the template strand.
Template?
Uh-huh. Which kind of makes sense, right? We're using it as a template to recreate the mRNA. It's kind of like what we're basing everything else off of. So this is our template strand.
What?
This is our template strand.
Okay.
Now we get to go in and match up all of these. What pairs with T? Hey,
You should like
Hey.
in
A.
the comments. Can I like comment my username?
please don't. Now this one's a little tricky.
My YouTube channel.
This one's tricky. What bonds with A?
T.
T if it's DNA. What about RNA? U.
I'm smart.
You are smart. So our orange kind of color is U. So now in DNA, we have T. In RNA, we have a U. We don't have any T's. C still is going to bond with G, C, G,
Literally it.
A, B, C.
I'm gonna look up Bailey Berman.
On what? Oh my god.
It's my school account. I mean, it's just videos. Most of them are unlisted. All right, so we're just going through. We're pairing up which ones would pair with which, right? G's are always going to pair with C's. That
Are
doesn't
they all
change.
marked safe for children? Oh wait, that's just
No.
your algebra. Never mind.
No, sometimes I can't mark them safe.
What
for
is your
children.
account called?
It's just my name. It's a school account. It's my school email.
So literally Bailey Berman.
Yeah. You probably won't see many because most of them are unlisted. Okay, so that's all of our pairs.
Ow. Okay.
We've got some more labeling over here. So this opening is a nuclear pore,
What in the fuck? Bro.
not a nuclear pore. A
I got
nuclear pore.
Slurp, slurp, Bailey the Berman.
That's not me.
It's a cat drinking out of a freaking water
Oh,
fountain.
yeah, Berman cats are like a breed of cat. So if you look up my name, you'll just
No,
get a
I
bunch of
got two
cats.
accounts here. They're both you, I think.
Maybe.
Baby.
That's not me.
No.
And then the space outside of our nucleus is our cytoplasm. So our nuclear pore is that opening that allows the mRNA to come out. And then the cytoplasm is where our next step will happen.
Did you find it?
Yeah. Using coordinates to compute perimeter and area, unit 2.
Yeah, those are all my geometry Admentum notes. You're welcome. Those are not unlisted. Now, how is RNA a little different to DNA? RNA has some differences. So RNA has a ribose sugar.
Dang, your bunch of friends like 5 years ago were crazy.
I was just like trying
Okay.
to think what's on there.
And then in DNA, we call this a deoxyribose sugar.
Wait, is that okay?
I know what you're watching. It was a video I had to make for my freshman year of college.
Wait, let me see.
We'll watch it later.
So nosy. Oh my god.
I know. Riley's trying to come to my apartment. I've got a stalker. Now, RNA also has a uracil base. So we have something called uracil. That's what that U stands for.
Is after the C, is there an I?
Yes.
Okay.
So we have something called uracil instead of thymine.
So you actually get views on these videos? Oh wait, I forgot what we were doing.
So we have uracil instead of thymine. So we have a U instead of a T. And then RNA is usually single-stranded. So it's only got one strand, while our DNA is usually double-stranded. So those are some differences that RNA has to DNA. It's got a ribose sugar instead of deoxyribose. It has a uracil instead of a thymine. And it's usually single-stranded, while a DNA is usually double-stranded. Okay. I'll leave this up for half a second.
Hmm?
Are you good?
Hmm? What's that one below cytoplasm?
Nuclear pore. It's the opening in our nucleus. One last page. Woo! This one also, most of the writing was on the first page. These ones are more just like pictures.
I do not want to be right.
Okay. With translation, we are taking our mRNA and we are converting it into a protein. So with translation, we are taking our mRNA that we just made, and we are now finally turning it into a protein.
Yeah, well, either way you
Now let's label our picture because we've got some stuff in here. Now this kind of T shape, right? It's shaped like a T. This is literally called tRNA. So that one I think is pretty easy to remember because it's shaped like a T. Like a T. So it's called tRNA. Now these little kind of circles that we're adding onto a growing chain, these are our amino acids. Right? An amino acid is what a protein is made out of. So these are all of our individual amino acids. Our amino acids are held together and bonded with a peptide bond. So our amino acids are bonded together with something called a peptide bond to make that polypeptide chain. Now on our mRNA, we said that these groups of 3 are called our codons.
Or what?
A codon. So every group of 3 is a codon. And what pairs with our codon, like over here, right, U will pair with A will pair with U, C will pair with G. This is an anticodon. So an anticodon bonds with a codon. This whole strand is our mRNA. And then over here, this big giant orange subunit, this is our ribosome. So that's what they would look like. Our ribosomes are usually made of like a smaller part on the bottom and then a larger part on top.
I thought
Then-
ribosomes were just little dots.
Well, they look like little dots in the cells, but this is what they actually look like if you were to zoom in.
Oh.
Mm-hmm.
We went into cells and now we're going even deeper.
Mm-hmm. 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 They're really tiny. And then at the very end of our mRNA is our stop codon. So this is what tells our tRNA to stop making the protein, and we're all done. Now let's talk about how translation works. So translation works by having our mRNA attached to a ribosome. And those ribosomes are in the cytoplasm. So our mRNA has left the nucleus. It's found a ribosome in the cytoplasm and it attaches to it. Now the ribosome lines up complementary tRNA molecules. So it gathers all of the tRNA molecules. And then their anticodons, so anticodons, must match the codons on the mRNA strand. So those tRNA ones have to match perfectly to the mRNA strand. With each tRNA, it's going to carry an amino acid. So all of our tRNAs have a specific anticodon, and they're also carrying with them an amino acid. So as they line up, the amino acids form peptide bonds between them, which builds our polypeptide strand. And that eventually will become a protein once it's all done. The tRNA molecules will detach from their amino acids. So once they've deposited that amino acid, they leave. They don't need that amino acid anymore. And then they leave to pick up new amino acids. So our tRNAs are really just carrying the amino acids. Once they deposit them on the chain, they go get more. So they're just carrying them to the ribosome and then they leave. All right.
Bailey, I won't be alone for algebra. Madison's coming.
Oh, good.
Because I convinced her.
Oh, good. I'm good.
No, I'm kidding. She just texted me and goes, I woke up late, but I'm on my way.
So when we read these three bases, our first one would be on the left column, our second one is on the top, and then our third is over here. So if I had something like UAC right here, and I need to figure out the amino acid that belongs to UAC, I would go over here. My first base is U, so it's going to be anything along this row. My second base is A, so it's going to be anything in this box. And my third base is C, so I'm going to follow C over. And so that amino acid must be tyrosine, which we shorten as TYR. Now our second mRNA codon is ACC, so same thing. I'm going to go down here. My first base is A, so it's going to be anything in here. My second base is C, so it's going to be anything in this box. Now, does it matter what the third letter is? If these are all THR? No. So it's going to be a C, but it doesn't really matter what the third base is. So my second is going to be this THR. And then my third amino acid is this GUC. So our G's are here, our U's are here, and then C is the second one, which is valine. Now, if we're looking at our DNA and we're trying to make our mRNA, we have to pair this up, right? So G pairs with C, C pairs with G, A pairs with U if we're making our mRNA. If we're making our DNA, A would pair with T. Okay, but I'm just matching them up. So A is going to pair with U's, T pairs with an A, and then ACC would become UGG. And then same thing, we can do our amino acids. So we're looking at CG at U. So C is all of this. G is always arginine, and then so U is also going to be arginine. So that we shorten to ARG. ARG. Our second one is UUA. So I look at U, I look at U, so it's going to be in this box. And then A, if you follow A all the way over, it's LEU, or leucine. And then our last one, UG. We'd go do U, G, and then all the way down here at the bottom, G, TRP. So that is how you would use this kind of chart to figure out the amino acids that would go with each of these codons. And then the very last thing that we have to do is we have to draw in our little missing anticodon. So here we have three bases. We have an A, a G, and a C. What is going to pair with our A?
Where? Oh.
Right here.
U.
U, correct. So I'm going to draw in a little U that will pair up with my A. I'm going to give it a little point, that way it fits right in there. What's going to pair with our G? C. And lastly, what is going to pair with C? G. So then to figure out what amino acid is going to be carried, we just have to look for UGC on our, or UCG on our table. So I would go over here. U, my second letter is C, so then my third letter is going to be G, and we have serine. So then this would carry a serine amino acid. And that should be all.